maybe something else. Um, yeah, but life is complicated, and there's no formula that describes biological organism other than this one. DNA is a very long string of letters. You could think of it as a long formula that contains instructions to make another living organism. Um, so if you look closely, DNA is made of nucleotides. So here's one. You have uh, a charged, negatively charged phosphate, sugar, and base. So base, that's something that is different across different nucleotides. ACGT, two strands come together to form a helix. And so the dimension of the helix are about 2 nanometers across, and the stacking distance is about zero point 0.3 nanometers. And yet, this highly charged polymer somehow fits within the nucleus. So there are 2 meters of DNA in each and every cell of ours, well, except for red blood cells. And yet, it fits into something that is only several microns across. And the reason how it happens is because they are ions. So we've been using our computational microscope to investigate various DNA systems. So very briefly, atom by atom, we build microscopic models of, say, DNA, and then we simulate processes by integrating Newton's equation of motion. So at the end of the day, we get this kind of a movie where things move. Um, my first example here will have to do with DNA electrostatics. As you all know, same charges repel in vacuum, but situation is different once you put the charges into solution. In solution, you can have pretty much anything from repulsion to attraction. In case of DNA, it's important because DNA is surrounded by water and ions, modulate the interactions between DNA. So experimentally, we know that for monovalent and divalent ions, generally, you see a repulsion between DNA, but once you go to multivalent ions, you could see attraction. Situation is a little bit different when you have DNA molecules confined to a surface, but that's a nuance. So we were really, actually, the reason we wanted to study DNA systems is that we wanted to simulate the virus, right? The antiviral particles. That was our original motivation back in 2009. And the first study in that kind of umbrella was to look at a system of many DNA molecules, so-called DNA bundles, right? Uh, so in this specific example, we have 64 of them. Each of them is connected to itself over purity boundary conditions. We add cations. The type and concentration of cations we can vary. So the water, and then the trick we do is we confine everything to fit into a cylinder of a certain radius, right? And and then we simulate, right? So the trick with confinement is important because we are able to measure how much force is required to keep the bundle in a certain confinement. And that is something that can be translated into pressure, and pressure is something that is known experimentally, right? So really, we wanted to first see what's the physics of DNA in bundles, and our first work in this area was a failure. Okay. So if you look at the DNA molecules and what happens to them as you simulate them, so it's kind of a top-down view. Each circle is a molecule. You can see that they start to cluster. And in the background, you're looking at local concentration of ions, and you can see that local concentration of ions can reach, well, almost 5 molar as the simulation progresses. So in experimental, we were expecting to see a nice hexagonal pattern, but that's not what we saw. Also, and the reason for that aggregation is contact pairs, right? So you would have an ion that would mediate interactions between the DNA molecules, and that is what causing the array to lose its hexagonal structure. Now, once we do comparison with experiment, we kind of see a disastrous result. So this is experiment over here, and those are our simulations, charm and amber. And this is pressure versus DNA distance, and well, we are nowhere near, so we are by factor 10 off, really. So disappointment, but it turns out that we understand what happens. The reason is really has to do with the force field. So the force field was never calibrated to reproduce quantitatively the interactions with ions. And so Jijun was a postdoc back then in my lab. Now he's a professor in Korea. So he went ahead and fixed kind of force field for a library of compounds using so-called osmotic pressure calculations. And this set of parameters is available on our website. If you can find it, email me. And basically, using independent experimental measurements, we could calibrate interactions. And with that, everything kind of is right on. So we can describe the behavior of DNA molecules in dense bundles. I believe this is a movie, right? So you can see that they on average form a hexagonal pattern. The pressure is right on, and yeah, so we kind of now have a quantitative tool to study the physics of dense DNA systems. All right. Now the next part of my talk will be about interactions between DNA molecules, and it's sometimes interesting to see how we end up doing certain projects. Now, in this particular case, it can be traced back to a conference I attended back in 2008, where I listened to Cornishev's talk on DNA telepathy, which was kind of crazy, to be honest, at first. So his theory predicted that if you have two DNA molecules, double-strand DNA molecules, so they kind of can feel their sequence through electrostatic interactions. So we decided to test it in the computer, and this is how we do it. We took DNA molecules, added ions, so we use spermine, which is +4 cation, so you would have a condensation of DNA under such conditions. We add background electrolyte, water ions, and then we just ask our computer a question, how much force is required to keep two DNA molecules a certain distance. So once we do it systematically, we are averaging out over all other degrees of freedom. We can find the effective force. We can integrate the force, get the free energy profile directly, which has a minimum, right? So it's good, meaning that there is a attraction overall. And what was surprising was that when Jijun repeated those simulations for DNA made of C and G nucleotides and A and T nucleotides, he found that the A T nucleotides they attract actually much stronger, which was kind of shocking. And we didn't believe it first, but then we looked closely and we couldn't find anything wrong with our simulations. It was also interesting that if you look at literature that describes how DNA is organized in biological cells, you'll find that AT-rich DNA typically form like clusters, whereas CG-rich they are open, so it was consistent with our calculations. The mechanism was actually not really physics, it was more chemistry. So the reason is that the thymine nucleotides, or T, it has this methyl group, which does not allow the spermine to bind to the groups of the molecules, and that kind of promotes their bridging interactions with other molecules, and that's how we end up with the attraction. So with that insight, we ask the question, what other systems have methyl groups? And of course, that has to do with methylated DNA. DNA, all our cells have the same DNA, right? But our liver cell, our hair cell, our skin cells, they all look different and behave differently because certain parts of the genomes are on and off. And one mechanism to turn genes on and off is to methylate DNA. So you have a cytosine, but then you add a methyl group, and then your gene becomes kind of silent. So We repeated our simulations for DNA containing methylated cytosine, and indeed we find that with methylation we see much stronger attraction. So that would have been a nice computational paper if it wasn't for the fact that we were at that time the same floor as T.J. Ha, who is like a god of single molecule threat. So his postdoc decided to prove us wrong and made a system where you have two DNA molecules like in a small lipid bubble, and the two molecules have two fluorophile dyes. So when they come together, they emit threat, and when they apart, there's no threat. And then we can make constructs that are of the same length but contain different sequences. And we can count how frequently molecules come together and how frequently they're apart, depending on their sequence. And satisfyingly, we found that indeed, if we have 80 rich DNA, then they would attract stronger. If we methylate DNA, it will attract even stronger. So that was a direct experimental confirmation of our predictions. I'm actually very proud of this work because we, A, fixed the force field, made it computationally accurate. B, we discovered something new, made predictions, we understood the mechanism, and then experimentalists confirmed it. So the story actually doesn't end here. We then showed that pretty much similar results happens when instead of spearmine, we use something more biologically relevant, like polylysin, which are the tails that appear in histone proteins. We also made a prediction that because of this differential interaction, DNA molecules would want to aggregate and form like microphase separation, where domains rich in AT would kind of cluster together and the CG-rich domains would not. So that was prediction back in 2018. And interestingly, those kind of things were recently confirmed. So there are two high-profile papers. I think this one was published in Nature eventually, again by TJ Ha and his lab. They looked at how nucleosome aggregate once we take them out of a cell. We fractionalize them, we condense them, and then you sequence what's condensed. And then they could actually say that indeed there is this physical forces that are responsible for organization of DNA in chromatin. And another paper, it's a completely different lab, looked at, showed that if you take an intruder DNA and you inject it into a nucleus, it will actually partition according to its sequence within the local environment. So, kind of complete story, prediction, uh, confirmation, and now it's like it's a phenomena that people study without our involvement. All right, so my second part of the talk will be about viruses. And of course, you all know that viruses are important. We're just out of pandemic. Who knows when the next one will come? We actually know quite a bit about viruses experimentally. We know the sequence of their genome. We know the structure of their enclosure, so-called capsid, right? So what you're looking at is this protein shell where the DNA is hidden. Um, but we really don't know much about what's inside of the virus because we just don't have tools experimentally to look into organization of the genome. Typically, when people talk about the structure of the genome, they show this picture. There's this concentric rings arranged there, and that's why people think that the DNA forms as loops. But in reality, we don't know. We we don't have the experimental means to look at the genome structure. And one motivation for us was that if indeed we can resolve the structure of the genomes, perhaps there are some drug targets that we can use. So, so we decided to determine the structure through brute force simulations. We decided to package DNA into the genome, right? This is a very difficult task because if you look at, so the DNA here is what's like 38 thousand base pairs, right? It is about almost a micron in length, and doing this in a atom MD is a mission impossible. We kind of really tried to see how we can do it in a atom, but I guess we weren't smart enough to figure out. So we had to invent a new method, and that's kind of what happened. Um, in our other line of research, we looked at the origami system, something that I briefly introduced yesterday. And the key insight was that we don't have to simulate everything at all atom resolution. In fact, we can start with something very coarse, and then as we progress, we can refine our simulation, eventually ending up with a fully atomistic model. So here's proof of principle. You're looking at a DNA molecule at same piece of DNA at different resolutions. The DNA has the same properties, and that's how we can describe very large systems. So this method has been, the multi-resolution method of DNA has been used to simulate various DNA origami systems like those jewelry boxes. Here's a system to model a capture of DNA plates and use it as a voltage sensor. All kind of gears, rabbits, flasks. So the field designed a lot of those systems, and we now gave the field a tool to simulate them, eventually arriving with atomically precise structure, which is something that the field didn't have. So that was our contribution. Now, but with a tool like this, we are able to simulate the process of DNA packaging. So we start with a experimental structure of the enclosure. We replace it by a field, and then we have a portal where the motor sits, and we package DNA just like that. So the DNA here is colored from blue to red. Blue is the part that is get packaged first, and the red is last. We had access to what was the best supercomputer back then. So you run multiple replicas, and you can see kind of already that looking at those 16 different configurations, is they're all different, right? So one big take-home message here that this concept of individuality, like we are all humans, very familiar with, extends down to very basic forms of life, like viruses. So each of the genome configuration is different. And the reason why they're different is because during the packaging, we introduce these large topological defects that constrain the system. Those defects cannot be resolved. Like for example, here you have a DNA molecule that protrudes from one side of the capsid to the other, and that gives capsids their individual features. Now, interesting fact was that the mechanism by which the DNA is packaged is in loops. So we call it a switchback loop, and you will see three loops being packaged. Like you see, there's a green one, and then it becomes too costly to package. So another one nucleates, and then another one nucleates, and pretty much whole of the genome is packaged in those loops. Now, but why can't we see those nice spools, right? Experimentally, we know that they should have those concentric rings, and in our mechanism, we don't. Well, the reason has to do with averaging. So uh, the picture on the left was obtained by averaging over 1,300 different particles. And if we do the same with our simulated snapshots, like there's no symmetrization, you can kind of already see like a little bit of a order at the edges of the capsule, but then if you add more and more and more and more, you average out configuration, so you end up with something that is pretty much concentric. We could also look at experimental images of the same virus. So this is HKM87 experiment, individual particles. You can also see there's this one order, and then there's like a protrusion there. You can find some of our configurations that look exactly just like that. So, but there have been several coarse grain simulations of DNA packaging. So in principle, it wasn't. I mean, we did it well, right? So it was a long simulation. Some simulations were a year long, but really, what is unique about this work that I'm also very proud of is that we went all the way to get the all atom resolution. So this is how we did it. So once we packaged the virus, we simulated it at different levels of coarse graining, increasing the resolution as we go on until our model was accurate enough so we can fit the all-atom uh, model of a DNA. So this is a fully atomistic model of the genome, which is 38,000 base pair, probably the longest covalent molecule simulated in all-atom resolution. And then we kind of equilibrated the ion atmosphere because we already roughly knew how many ions we need to put for the system to be stable. And then we put back the capsid and surrounded by water ions. And that's how we got the first complete all-atom structure of a package virus. So we again had access to Frontera, so we built several models, ran it. It was an expensive simulation, but okay, someone has to do it, so it was us. All right, so what did we learn? And I really don't have time to go through all the interesting stuff that we learned. I just give you kind of one example. But in general, there was a wealth of information. Electrostatics in those systems is very, very, very specific. We figured it out eventually, but kind of took a year to figure out why exactly we see what we see. But this example is intuitive. So the DNA is an electrostatic bomb, right? It wants to explode, but it wants to do it in a spherically symmetric way, right? So it's repelled in all direction, but the enclosure is not spherical. Enclosure has this icosahedral symmetry, right? It has edges, faces, and vertices. So we were wondering, what happens to the genome as it tries to fit into the confinement of the capsid. So for that, we looked at the organization of the DNA at those features. So for example, we found that the DNA crosses the edge of a capsid roughly at 45 degrees. There's a number of reproducible topological defects at the interface of the genome and the capsid, like the triangular defects and so on. But the most striking feature was this observation that this DNA, it kind of protrudes into the vertices of the capsid. There is a like empty space there. So you're looking at the genome that is colored by the distance to its center. And those snow-capped mountains, so to say, is a feature that emerged during our microsecond simulations, highly reproducible. So we published this, and then someone pointed out that actually those things were observed in experiment. So you're looking at another double-strand DNA virus, herpes virus, which is a virus that affects humans. And if you look closely, you can see that there's this protrusions of DNA into the vertices of the herpes virus uh, closure. So in a way, we were able to. I really actually didn't know about this when we published the work. It was just kind of post thing. So with this work, we show that we can use multi-resolution method to predict the structure of the genome with atomic precision. So we of course now work on this and model other viruses quite a bit. Now, I'm gonna spend maybe just a few minutes, so I want to get some questions, just reminding you what I talked about yesterday about electromotors. There, our key discovery was that the DNA itself, I'm gonna skip this, that the DNA itself is a pretty good motor, right? That it can spin when we apply electric field. And the way we do it is by an experiment. We place it into a carbon nanotube connected to graphene, something like that. So if you apply electric field, it would rotate on average in the direction that you would intuitively expect it to rotate. Now, one thing I didn't talk about yesterday was the following, that with all-atom electrodynamics, we can measure effective forces and torques. And this is exactly what we did here. With forces, you just restrain the molecule using springs, and then if the molecule displaces, you can compute how much force it experiences. So we can do the same with torques. So we apply electric field and we constrain the DNA molecule so it wants to like rotate, but because there's springs, it rotates by a little bit. And from this displacement, we can tell exactly how much torque is applied to the DNA molecule. So once we knew that, we did the following computational experiments. So we took a DNA molecule It's going to go through a nanopore here, and if you look closely, I'll try to catch it like this, right? Do you see what happens? There's like if you ever like take a wire and start twisting it, if you do it strong enough, you'll see that it will form those kind of arrangements, which in biology are called plactoniums, right? So you'll see plactonium forming on the DNA molecule as it passes through nanopore. So that was our prediction that we described in this 2023 work. And I'm happy to report that that's something that was experimentally observed by Ulrich Kaiser lab in Cambridge with Christian at CISA. So they did coarse-grained simulations. Our contribution to this work was to predict how much torque the DNA would see, and experimentally they see features where, you know, three strands pass through the nanopore, and that was later interpreted. So previously people thought that those were knots in the DNA, but as our work shows, those are actually plactoniums. So again, kind of went the full circle with this work. We showed that nanopores can be used as molecular motors, electromotors. They can generate torque, and our prediction that DNA, the torque in DNA will produce plactoniums was confirmed by experiment in this recent work. That's pretty much all that I wanted to tell you today. This is my lab at some point in time. I'm privileged to be working with a very talented group of people. We've been funded by various agencies, and of course, our main resource are supercomputers that we have access to. And I thank you for your attention. I maybe can have a few questions if you have them. Thanks.
Thank you very much, Alexei, for this incredible journey across DNA simulation. It has been very interesting. I have some questions, but first I want to hear from the people in the room if they have any question.
Do we have any questions? Please ask if you have. Alexei, probably I'm sure that we have a lot of questions because this presentation is really amazing, and we know most of your papers, and I would like to ask you about um, simulation of viruses. You mentioned that this is electrostatic bomb, and actually you mentioned this formulation in PhysRev X with Kornyshev you published,
Right.
and yes, I like a lot because in fact this is true. DNA is electrostatic bomb, and if you construct the virus, construct DNA inside the capsid, you have you should have a lot of counterions inside because it will not stable. And how many ions you add, and how you determine this amount of ions?
Yeah, that's an excellent question. And this is actually why we did this work to start, right? Because there's a lot more ions than outside, right? So in this kind of work, we know how much ions we put outside. So it's a called bulk concentration. And then the ions, they can diffuse through this confining wall. We did the same trick for the virus, by the way. We kind of confine it in a semi-permeable membrane so the ions can equilibrate. But from this work, we kind of knew roughly how much we need to add. For a mixture of sodium and magnesium, they were equally split in terms of charge, but then the amount compensates the charge of the DNA. So that's kind of roughly was our guess, right? That was our initial guess, and it was not very far from what we saw, because then we allow the ions to kind of diffuse through. Ions water can exchange, but DNA cannot. So in a way, it's an equilibration simulation, and that's how we get the ion atmosphere, quote unquote, correctly within the virus. Yeah.
Thank you very much. Other questions? Dr. Chernyak, please.
Thank you. I'd like to ask about electromotor, and in particular, I'd like to know, are there any minimal electric potentials to provide a stable net spin rotation of this DNA molecule, DNA, well, part of
Right.
DNA.
Yeah, so in this example, you apply 200 millivolts, which is very common voltage, and that
Okay.
is sufficient to generate sustained rotation at almost a billion of RPMs for a small piece of DNA. Now, but this simulation, this is the one where we restrain it, so it can't really move. But the one that I showed yesterday, I think it's somewhere here, like this one, this one was not restrained at all, so it's free system. There we applied 400 millivolts, and if you look at
Okay.
what my colleagues do in experiments, they apply about 200 millivolts. So it's standard voltage range for nanopore experiments.
Okay. And another question, are there any possibility to provide this not in graphene coating, but in free environment, like just DNA nanomolecule and applied at some direction electric field, and it rotates or
Yeah, so that's a good point. The problem with that is that you need to apply very high field. What nanopore does, it focuses all of the field to the nanopore. So even though you apply field away from the membrane, because it's a conductor, all of the field lines are kind of condensed to the nanopore. So you have very high electric field in the pore, and that is what you need to spin the DNA. If you were to do it in a free solution, it probably won't work because you won't have enough field. What you could do in free solution, you can try to flow water. And actually,
okay.
I don't have time to talk about it, but you can also imagine that if you just have a flow of water, it would also rotate the DNA, and that's what we've shown also in simulation and experiment. So that kind of method probably would work better for free DNA. Yeah.
And the final question, are there any dependent on salinity of water to rotate this DNA, or it is independent?
Mm, Kind of excellent question. We killed 2 years doing this, and of course, don't have time to go over it. But experimentally, we found that if we repeat this DNA turbine at different salt, it spins the other way. Okay,
Okay.
that was like total shock, and it took 2 years for us to figure out why. The nice thing is that if we repeat simulations at higher salt, we also see it spinning the other way, right? So it's kind of amazing that it works. Like it's a model, right? But we can capture essential physics of DNA in solution. But then it took us 2 years to figure out why it spins, and it's not easy. Like the explanation is not intuitive. One thing I learned from this is that we humans don't have intuition for Navier-Stokes equation at all. Electrostatic is okay, but Navier-Stokes is such a complex 3D thing that we figured it out, but yeah, it's in the paper. It's complicated. Yeah.
Okay. Thank you. Thank you.
Francesca, we still have questions in our conference room, but I think we can switch to Zoom, please.
Okay, I see Galina has raised her hand, so please, Galina.
Can I ask?
Yes, please.
My question is about condensation in viruses. You mentioned that is it similar than in nuclear or it is different? And also how to imagine this process.
Yeah, that's an excellent point.
These.
I mean, the only thing that is similar is the DNA, right? In nucleus, you have you have those histone proteins that assist the process of condensation, and also ions and spermine at some quantities. In viruses, the the reason why DNA viruses have DNA inside is because of the motor, right? So it's not spontaneous process. You actually have a motor protein that consumes energy to package the DNA inside. The ions are there to make it possible, uh, because without the ions it would just explode. But once you add enough ions, it will compensate the charge of the DNA, so that the virus can work against it. So I would say this screened interactions between DNA. The role of counter-ions is, of course, important for this. The mechanism of arrangement of those loops, it's also seen in nucleus, surprisingly. A nucleus is thought to be organized by motor proteins that extrude them into loops. Similarly, we saw in our systems of packaged viruses that the DNA is arranged also into loops. The loops are different scale. So in our case, in viruses, it's like 150 base pairs. In nucleus, it's like you're talking about kilobases of DNA. But perhaps, loops is a way you deal with highly charged polymers. I don't know. Maybe as we study more systems, we can generalize whether or not it's something that is a common pattern, or maybe that was just a coincidence.
Okay, thank you.
I don't see any other hand raised remotely on Zoom.
Yes, we have in conference hall.
I have some questions.
Okay.
I will. I have many questions, but just a few. So, in the modeling of coarse grain modeling of the packaging of the DNA in the virus, was sequence specificity somehow taken into account, and do you think it might have a role?
Yeah, so the coarse grain part is not sequence aware, right? So it's a polymer model. Once we map it to all atom resolution, of course, it is a sequence of the HK97 genome, right? It has it's like exactly the same experimental sequence. So whether it plays a role, I don't know. We thought about it for a while. Experimentally, it's known that for this specific virus, it doesn't matter. Because there is a way to cross-link DNA to the capsid and then tell, is there any preference in terms of sequence, what's where, and as far as I know, there wasn't, right? So that was kind of gave us support to not taking sequence into account in coarse grain modeling, We also looked at our simulated all atom structures with regard to any kind of sequence preference. We didn't really find anything. But again, that's not to say that it doesn't exist. Perhaps we just don't have enough data
Yeah, I'm
points.
surprised. I would expect a little bit. Okay, in the calculation, probably you cannot see so much because it's already formed once the loops are already formed when you go up
Right.
to the atomistic level. Because I would expect, for example, a tract to be deflected from loops. So I would expect the sequence to somehow determine where the loops form, or maybe better, they don't form. So I would be, I don't know.
No, you're right. Yeah, your intuition is correct. That was actually shown for other system, like for plaquetines, for example. It was a very nice paper from Kees Dekker's lab, where they showed that, this tip of the plaquetinum, if you see my cursor, that wasn't very sequence specific. You need to have a certain sequences that you typically find at the tip of a plaquetinum. And, with that, it's conceivable that once you package the virus, perhaps, those loops will format as predefined places in the genome. Yeah, that's possible. Yeah.
I mean, all the work you have been doing is amazing. I have just a little question, just a curiosity. I guess it's very fast. For the periodicity of the system where you have a 65 DNA packaged in a circle, and you have the periodicity also along the DNA axis, how do you deal with that? Because I see that they are free to move, but I don't know. I am.
You mean like this systems?
No. The simple system, the simpler system that you showed
This one?
before, where you have the ions that can diffuse inside the system. No, it was
Oh, I know this one.
exactly
Yeah.
page 7. Yes.
Oh, yeah. So yeah, it's so we use periodic boundary conditions to our advantage. So what we simulate is infinite DNA. If you
Yeah,
think about it.
you are somehow linking a little bit.
Yeah, it's so there is a patch, and you can. Well, I mean, it's not. I'm making it simple. It's a little bit complicated because you have to choose the right length of the DNA. So it actually matters. Sequence dependent, right? But we kind of knew roughly what to choose. And then you link it on itself and you have like effectively infinite DNA, which is great. So you don't, they're always kind of parallel to one another, right?
Yeah.
And then well, it's calibration is a bit tricky because you have to fix the length of the system along Z, but you allow it to calibrate in X and Y, so you get the right density and pressure. So it's, I mean, it's not complicated, but it's a little bit involved. But once you do it once, you can do it many times. Yeah.
Yeah. I mean, this is some question because we also did something with Sergi about it similarly, but we decided not to put the periodicity in there, so to allow the molecule uh, to curve if needed by the interaction with the ions, because in this way we thought since the DNA is not so long, if we periodically bind it to have that length, we are maybe hindering some other movement. So that's why we try to follow other ways, but it's complicated. But anyway, for you, it's out. That the phenomenon is not so relevant, I think. And anyway, we don't have so much time. Thank you very much for your talk and for
Thank you.
your answer. I don't know, Sergi, if you want to give some more time for the question in the room.
Yes, we have questions. We have a question in the conference hall, please.
Thank you. Thank you. My question is about DNA condensates. See, in condensates, DNA molecules are quite dense, and they form kind of liquid crystal phases like kinematics, and it's possible to form like smectic phases or holosteric phases in DNA molecular systems.
I don't know if there is a problem with the microphone.
Yeah.
I cannot hear very well. I heard very fragmented.
I can repeat, sorry. Thank you, Aleksei. It's an inspiring lecture, and my question is a question about DNA condensates. They are such systems are quite dense, and they form like similar something to the nematic phases, like in liquid crystals, right?
That's right. Yeah.
They are ordered rotationally. And is it possible to observe in such systems other phases like smectic phases or holosteric phases?
That's a really good question. I guess if you're interested in this area, you'll find this because we see it in packaged virus, right? So what you're looking at is this nematic order, right? So you have this very nice local nematic phase, but then you have a topological defects. In fact, I didn't have time to look at it, but you can do like this Frank-Cosin distortion energy calculations. You can see where the defects are. So this is methodology of liquid crystals, really. So for answering your question directly for those things, if you have short fragments of DNA, and that is something that Tommaso Bellini showed very nicely in his work, you could have spectric phases, because there you'll have also end-to-end interactions. So in this case, we don't see it because it's an infinite DNA, but if you break it into smaller fragments, you will see them arranging into all kind of liquid crystalline phases. And yeah, it's experimentally known. Reproducing it in simulations. I think there's a group in Italy who did that some time ago. But yeah, definitely very rich system to see liquid crystal-like transitions.
Okay, I see you. Thank you, Alexei.
Thank you very much. Are there any questions in conference hall? Do you have? Please, if you have, please ask. If not, we go forward.
Hello, I would like to ask if the connection between ionic state inside the capsid and release of DNA. So I mean, if ions, for example, condensation change the DNA release from the capsid of virus.
Yeah, we didn't really study the ejection of the DNA from capsids in all-atom method. We did it with a coarse-grained model. I don't know. People still arguing how exactly ejection happens and what role ions play. There's some people think it's that this you have a gradient of concentration inside and outside, and that somehow creates a water flow and that pushes the DNA out. I don't know if it's true. We haven't yet simulated it, but that's on our to-do list. Yeah, that's kind of all I can say about your question, if I understood it correctly. So, yeah.
Yes, you understood correctly. Thank you.
You're welcome.
Thank you very much, Francesca. So.
Okay, so thank you again, Aleksei.
Sure. Thanks so much.